everyone. My name is Ryan Miller. I'm part of the Kong group. Today, I'm going to be talking about antioxidant regulation of mesenchymal stem cell senescence with the aims to restore their therapeutic potential for the treatment of neurological disorders. So mesenchymal stem cells, or MSCs, as I'll call them in this talk, have widely been used in the fields of tissue engineering and regenerative medicine due to their differentiation potential. But as Will mentioned earlier in his talk, groups are now looking at direct delivery of the MSC secretome as opposed to implantation of the MSCs to restore normal physiological function. So one of the studies have been to try to decouple what is in the MSC secretome. Some of the discoveries were that the exosomes released from MSCs contain an abundant amount of microRNA and messenger RNAs, and that they're also contain a multitude of growth factors and immunomodulatory factors as well. And there's been promising results by harvesting these MSCs from patients. This can be from adipose tissue, like my case, or it could also be from bone marrow. But when you're trying to harvest the secretome, you need a significant amount for these therapeutics. And the issue is that it's been well reported that culturing of these MSCs for long periods of time can lead to age-related cellular senescence, as well as oxidative stress-dependent cellular senescence as well. And this can lead to a reduction in their exosome production, a reduction in the growth factor secretion, and also a reduction in the immunomodulatory factor secretion. So what groups have been trying to do is prime the cell culture media with antioxidants. And these have good bioavailability if they use hydrophilic forms, but the issue is that the lifetime is relatively short. So our group asked, can we, instead of using a soluble form of an antioxidant, can we add monodispersed polymer stabilized antioxidant crystals that can extend control of the cell's oxidative state? And if so, does maintenance of the cell's oxidative state restore healthy paracrine expression? So the way, this is going to be brief, but the way that we formulate these monodispersed crystals is using droplet microfluidics. So you have these single emulsion drops, which if I lower the temperature in, I can get N-acetylcysteine, which is our antioxidant, to crystallize. And if I thermocycle these drops 5 to 10 times, I can get 1 crystal per drop, leading to very narrow crystal size distributions as seen in the histogram on the right, compared to a bulk thermocycling approach, which has a lot broad size distribution. I further want to note that these crystals are stabilized with hyaluronic acid conjugated with dopamine. This facilitates the longer extended delivery or release, as opposed to if it was pure crystalline form, it would dissolve in 90 minutes. And you can see the release curves in the bottom right. Two things that I want to note is that we have the prolonged antioxidant release. We suspect that this will extend the control of the oxidative state of the cell, and then also the highly uniform release due to the crystal size distribution should lead to less fluctuation in the intracellular ROS levels, because even micromolar fluctuations have been shown to lead to drastic changes in the cellular dynamics. So moving on to the in vitro models that I'm using. So as I mentioned, I'm using adipose-derived human mesenchymal stem cells. First, I want to introduce senescence into this healthy population of MSCs. So the way I do this is over the first 5 culture periods, I'm doing 25 micromolar injections of hydrogen peroxide each day. And then after those 5 days, I then do 6 days of treatment of 200 micromolar NAC crystals. And I'm adding the crystals every 2 days when you would have conventional cell media changes to just try to argue that this is a very easily implemented additive to culturing conditions. So I measured the degree of senescence in 2 ways. One is with beta-galactosidase. It's the protein that is overexpressed in senescent cells, specifically in the ribosomes. And you can see the histological stain in the optical images. The cells that have this really bright blue dye is going to be considered beta-gal positive. And in the bar graph in the bottom left, you can see that if there's no treatment, the fraction of beta-gal positive cells is about 90%. but with treatment of the microfluidic assembled crystals at day 6, I'm able to reduce it to 20%. I also want to further validate the degree of senescence, so I used qRT-PCR to quantify the gene expression for P16. This is the tumor suppression protein. It's also upregulated in senescent cells. And again, you see a very similar trend for the microfluidic crystals at day 6 for the treatment, led to a 1.5 times reduction in P16 gene expression. So I mentioned that the whole point of using an antioxidant is to control the oxidative state of the cell, so I really need to prove this point. So the same exact protocol to induce senescence and then out treatment with the crystals, but now I'm using a cell stain called CellRox green. This becomes oxidized by reactive oxygen species in the intracellular space and emits a bright green fluorescence. And I want to point out first in the confocal images that for the no treatment condition, you can see that the nuclei is much larger. This is a characteristic of a senescent cell. And then also that the green fluorescent intensity signal is significantly higher, and there's these bright green puncta that you can observe in the cytosolic space. I don't have the results in this presentation, but I'm doing co-localization, and it appears that these are co-localized in the mitochondria as well, which is also another marker for senescence of the mitochondria that release ROS. So when quantifying the intracellular oxidative stress levels, you can see again, similar trend that at day 6, this is for normalized intensity. So 1 is for a healthy cell, that you can see at days 4 and 6 that the microfluidic crystals have no statistical difference between a healthy MSC, where you have really high levels of oxidative stress in the senescent cells. So that's great that I can pull cells out of a senescent state. I want to add a caveat that if there's DNA damage, the cell will not come out of a senescent state. If it's oxidative stress driven senescence, you can pull them out of the oxidative state. But now I want to show that their functionality has been restored. So I mentioned 2 things: the exosomes as well as the characterizing expression of MSCs that's useful for therapeutics. So the first thing that I characterized is the exosome release, and I use nanoparticle tracking analysis as a DLS approach to quantify the total exosome count. And you can see after 5 and 6 days of treatment, the microfluidic crystals are able to restore normal, healthy exosome secretion, where the secretion for the senescent population in a treatment group was half of what it would be expected to be. I also characterized the paracrine expression of vascular endothelial growth factors, so that's VEGF. IGF is insulin-like growth factor, and IL-10 is a pro-inflammatory cytokine, interleukin-10. And for all 3 of these, the gene expressions, again, using qRT-PCR, you can see that I'm able to, with the microfluidic crystals, achieve baseline levels that are comparable to that of the normal, healthy, functioning MSCs, where there's deviations for the senescent populations. So with this, my future work is to extend the results that are presented today to a senescence propagation model. I have 2 models in mind. The first one that I started with was isolating the 2 different groups using a transwell. So in the upper well, I have senescent cells, and I have healthy MSCs in the bottom of the transwell. And through the membrane, the secretome can pass and show that senescence propagation occurs. And then I want to treat the healthy cells to show that I can block propagation. This has been promising, but I also am interested in the kinetics. So now I'm working on seeding the senescent MSCs in a center region and then having healthy regions at different concentric circles and seeing what the propagation rate of senescence looks like, and then treatment with the crystals. And then finally, as we understand the kinetics a little bit more, we would like to show that we can expand this cell line specifically derived from human patients and harvest the secretome and then test with in vitro models using neural stem cells to see what their differentiation potential looks like. So with that, that concludes my talk. I want to thank my main advisor, Dr. Hyun-Jun Kong, and the lab members. And I'm also co-advised by Dr. Hee-Sun Han in the chemistry department. And it's her lab that helped teach me how to fabricate these microfluidic devices, and all the microfluidic work comes from. And I also want to thank MBM for the opportunity to present this research and conduct this research. So if there's any questions, I'm happy to answer. Yes.
So when you're when you're adding those crystals to the culture, are they still in those microfluidic droplets?
No, so I break the drop after I thermocycle to get to a uniform size. Then, because they're decorated with hyaluronic acid dopamine, it actually drives the crystals to aggregate together. And this is what it allows me to have the extended release. So I break the droplets with an anti-surfactant. It pulls the RAN surfactant that's stabilizing the drop, then do washing steps, and then I add a suspension of the crystals to the cell media.
Okay, so they're just like floating. Do they get cytosed? Do they go inside the cell? Or?
Maybe as they dissolve to much smaller sizes, but the ones that I was showing there, the average diameter was about 10 micron. I would not expect that to undergo endocytosis, but if it enters the nanoscale, which I can't resolve under an optical microscope, there is potential. But the reason I use NAC to begin with is I wanted to be internalized to produce GSH, which Miles talked about, glutathione, which works in tandem with glutathione peroxidase.